entertaining a lot of perhaps wild goose chase hypotheses, we could actually cut through the Gordian knot and be able to get to the heart of the matter and figure out what's going on in these patients. So that really appeals to me. And just for those of you who aren't familiar with FMS, there could be such people in this audience, just a few images that perhaps will illustrate the features of FMF. So just by a little way of background, the attacks of FMF usually last on the order of 1 to 3 days. That's actually quite important in the course of this talk because we'll be talking about patients that have other periodic fever syndromes where the duration of the attacks is much different. So remember that. 1 to 3 day attacks. It's usually seen in individuals of Jewish, Arab, Armenian, Turkish, and Italian ancestry, some of the clinical features are shown here. One of the things that these patients frequently have, which our patient that I just told you about did not have, is episodes of peritoneal inflammation. And these are just air fluid levels on the upright film of the abdomen. These patients can also have pleural inflammation. They can have pleural effusion. Rarely, they can have pericarditis, and you can see, in this case, posterior pericardial effusion. Arthritis, as I mentioned, is a common manifestation. It's usually a non-deforming arthritis, although prior to the advent of colchicine treatment in FMF, chronic arthritis of the hip was seen in some patients. And then there's a rash that's called erysipeloid erythema, a reddish, raised, fairly well demarcated rash that usually occurs on the dorsum of the foot, or the ankle, or the lower leg. Histologically, what you see is lots of polys, as I mentioned in the case of our positive patient. And these patients sometimes can develop systemic amyloidosis. This is basically due to the deposition of a cleavage product of serum amyloid A, which is one of the acute phase proteins that's made during the inflammatory response. And it deposits in a number of vital organs, including the kidneys and the gastrointestinal tract, the lung, the liver, the thyroid, rarely in the heart. So in any case, I did get interested in positional cloning, maybe not immediately after seeing Sarkis, but within a couple of years of that. And so just in one slide, I will summarize a little bit of that adventure. So I did end up, through my fellow friend, making a collaboration with a group in Israel, this guy here, Dr. Mordecai Cross, who ran a very large FMF clinic in Israel. And so basically we did, during the summer of 1989, travel the countryside of Israel, going to people's homes, and after obtaining informed consent, which is in this notebook there, we would have the patients and their family members roll up their sleeves, and we would get blood samples, which we would then make Epstein-Barr virus cell lines from, which at that time was necessary to extract DNA. In any event, we did have a fairly large panel of families, and as you can see here, maybe you can't see, but some of them are consanguineous. In point of fact, this picture here, which has been modified for HIPAA purposes, does illustrate a consanguineous family. Parents are first cousins of one another, and there is a marked intrafamilial resemblance. In any case, we were able to map the gene ultimately to the short arm of chromosome 16 near the hemoglobin alpha locus. And then we sort of became the local chromosome 16B genome center at that time, because the genome project had not yet taken on the global dimensions that it ultimately did. And so we sort of became responsible for developing maps of this region of chromosome 16. First genetic maps and then physical maps of this region of chromosome 16. Narrowed the region down to a region that was about 200,000 base pairs, which turned out to contain 10 different genes. And as luck would have it, it was the 10th one that we interrogated that turned out to have the mutation associated with disease. And that turns out to encode what was, at that time, a novel protein that we called pyrin, after pyrexia, for fever. Now, it turned out that there was a French group that was also competing with us at that time, who fortunately found the same gene, some of the same mutations in that gene at the same time. They chose a different, more erudite name for the protein, the encoded protein, namely Mare Nostrum, after Mare Nostrum, for the Latin for the Mediterranean Sea. But whether you call it pyrin or Mare Nostrum, it turned out to be a novel protein. And of course, that was really what we were hoping for all along, was that we would learn something rather than at the end it would turn out to be IL-2 or something like that. And people would say, well, why did you spend however many years it was looking for the IL-2 gene in these patients with FMF? We could have told you that anyway. But it turned out that it was something novel, and it turns out that in fact the N-terminal domain of this protein is a domain that's found in some 20 different human proteins that are involved in the regulation of inflammation and apoptosis. And it is called the pyrin domain, not the Mare Nostrum domain. And basically, it assumes a 6-alpha helix structure that allows the formation of a dipole. And you can see that positive charges are denoted in blue here, the negative charges in red. And basically, pyrin domains can engage in cognate interactions with other pyrin domains, just as, for example, DEF domains can interact with one another in the TNF signal. So in any case, in the case of pyrin itself, it interacts with an adapter protein that's denoted ASC, for apoptosis-associated speck-like protein with a card domain, which is why we usually just refer to it as ASC. It has both an N-terminal pyrin domain, which interacts with pyrin, and a C-terminal card domain, which then interacts with the card domain of caspase-1, also known as IL-1 beta converting enzyme. So this is actually the protein that activates pro-IL-1 beta, a 31 kilodalton protein in humans that's the pro form of IL-1, into the biologically active 17 kilodalton form that mediates fever and inflammation. So then this does put pyrin upstream in that cascade, and at least gives us a beginning of an understanding of how pyrin, mutations in pyrin, might lead to unexplained fevers. In any case, it turns out in later work from our lab that in fact the C-terminal domain of pyrin, the so-called B30.2 domain, interacts also with the catalytic domains of caspase-1, shown here, the P20 and the P10. So there are multiple interactions there. And actually, that C-terminal B30.2 domain of pyrin is where most of the disease-associated mutations reside. And so that's really a very interesting thing. And it turns out that if one looks at DNA fingerprints associated with those mutations, that one can deduce probably common ancestors for the modern-day carriers of those mutations. So that, for example, the blue here depicts the M694V mutation, the substitution of valine for methionine at position 694. It's very common in the North African Jewish population. And in fact, that mutation with an associated DNA SNP haplotype is seen in other populations, such as Turks and Armenians and Iraqi Jews. And there's a fairly large stretch of DNA that's in common amongst them, suggesting common ancestry. Moreover, the carrier frequency for these mutations, and there are several of these mutations for which this holds, is extraordinarily high in Middle Eastern populations, on the order of 1 in 3 to 1 in 5 in those populations, which, given the fact that there are multiple mutations at these high frequencies, really suggests that there could have been something, some perhaps pathogen, that was selecting for these high carrier frequencies over the course of human history. So in any case, and if one models the B30.2 domain, it actually turns out that several of the mutations form a ring around a putative binding pocket for something. So that has been very interesting to us. So what could the selecting pathogen be? Well, we don't know, at least at this point. One speculation that I was discussing with a friend in Turkey a couple of weeks ago is that it might be smallpox. And And actually, the evidence for that, although speculative, is at least interesting. So there's been a lot of talk about where did smallpox first arise. It's an obligate human pathogen, and there's at least some archaeologic evidence, for example, that suggests that one of the early places where smallpox arose was